Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic in the series of food preservation, and the topic that we are going to discuss is ultrasound is used as a preservative. So basically the preservatives are what? Preservatives are the substances which is used to extend the shelf life of the food product. Preservatives are what? It is the substance which is used to enhance the shelf life of the food products. Okay, so you can use a preservative like in a chemical form, and sometime you can use it as a ultrasound waves. Okay, now we have to discuss that how these ultrasound waves affected the shelf life of the food product. Okay, and how it can affect it, like it eliminate the microorganisms present into it, eliminate the microorganisms present into them. Okay, when the elimination of the microorganism take place, the ultimately the shelf life of the food product are increases, and along with the elimination of the microorganisms, it also affect the chemical reaction of the food product or enzymatic reaction. So these are the important factors that we have to discuss, that how this ultrasound affect the shelf life of the food product, or you can say that how we use it as a preservative to enhance the shelf life of the food product, and along with the shelf life it also have a effect on their flavor, aroma and taste. So let's start the lecture. So the content that we are going to discuss is introduction part, ultrasound generation, use of ultrasound in food processing, ultrasound in food preservation, effect of ultrasound on food quality, then conclusion, and then your references. So the first topic that we are going to discuss is about the introduction of the ultrasound. So we all know that this ultrasound waves are what? They are very similar to the wave that generated from the sound. But here the difference between the ultrasound wave and the sound wave, that the frequency frequency is at above than 16 kilohertz. Ultrasound frequency are above the 16 kilohertz. That's why it cannot be detected by the human ear. Okay, these ultrasound waves refer to that the sound wave mechanical vibration which propagate through the solid, liquid and gases with the frequency greater than the upper limit of human hearing. Okay, we all know that we cannot hear this 16 kilohertz. That's why we are not hearing the ultrasound waves. It is very similar to that sound wave. But here the difference between the sound wave and the ultrasound wave is that their frequency. Ultrasound wave have frequency above than 16 kilohertz. That's why it cannot be detected by the human ear. These ultrasound waves are referred as a sound wave mechanical vibration when it propagates through either the media like your solid, liquid and gases, but their frequency is greater than the human hearing upper limit. We use our ultrasound in food processing, which includes the extraction, drying, crystallization, filtration, defoaming, homogenization, and it can also be used as a preservative technique for the preservation of the food product. We can use ultrasound in both type of the technique, like either you can use in a food processing like the conversion of the raw material into the food products, that is the food processing. Okay, which included your extraction, drying, crystallization, filtration, defoaming and homogenization. But along with this food processing, this ultrasound waves can also be used in a preservation technique. The main principle of this technology is that we have to reduce the processing time. When we use a ultrasound for the food processing, we reduces the processing time, and also energy will be saved, and it improves the shelf life and the quality of the food products. That's why we uses the ultrasound for the preservation and the food processing. Next is your, this is a frequency ranges of the sound. Okay, like human hearing, we can human can hear the sound at the range between 16 hertz to 18 kilohertz. Then conventional power ultrasound, that is 20 kilohertz to 100 kilohertz. Then extended for special application, that is your 20 kilohertz to 1 millihertz. And diagnostic ultrasound is in the range between the 5 millihertz to 10 millihertz. So these are the frequency range of the sound that we uh, that are present in the environment. Sound waves can propagate parallel. We all know that the sound wave can propagate the parallel or perpendicular to the direction of travel through the medium. Suppose that this is your medium. Now these, if the sound wave are travel, so either it can travel parallel to this, or either it can travel perpendicular to the direction of that media. That's why the propagation of the sound wave is normally which is associated with the liquid media. Okay, if the wave are the parallel, then it is known as a longitudinal wave, and if the wave is a perpendicular, then it is known as a shear waves. Okay, if wave is perpendicular, so then it is known as a shear wave, and if the wave is parallel, then it is known as a longitudinal wave. So these longitudinal wave have a capability to travel in either of the media, like it can be travel in a solid media, it can be travel in a liquid media, or it can be travel in a gaseous media. But longitudinal wave have a short wavelength as compared to that transducer dimension, which produce a sharply defined beams and high velocities. Okay, this diagram represented the longitudinal wave because it is travel just perpendicular to that media. Now, the next is your shear wave. Okay, so these shear wave have a particular motion which is perpendicular to the direction of wave propagation present in liquid and gases, but it cannot be support the shear under that normal condition. Shear wave only propagate through the solid media, and the velocity of that shear wave is very low as compared to the longitudinal wave. Okay, it is low as compared to that longitudinal waves. Ultrasound generation. So how these ultrasound are generated? So ultrasound can be generated by the system which contain the generator, transducer, and the application system. Okay, when we have to generate the ultrasound, then for generation of their ultrasonic wave, we required a system which contain three parts. The first one is your generator, then transducer, and the last one is your application system. Generator is used to produce the mechanical energy, because without mechanical energy, ultrasonic wave production is not possible. So we use just this generator for the production of that mechanical energy. Then the second part is your transducer. Transducer is used to convert that mechanical energy into the sound energy, and the frequency of that sound energy are the ultrasonic frequency. So generator generate the mechanical energy, and transducer convert that mechanical energy into the sound energy, and the frequency of that sound energy is the ultrasonic frequency. So we can use a three type of the transducer. The first one is your fluid driven, then magnetostractive, and piezoelectric. So these are the three type of the transducer which we use for the conversion of the mechanical energy into the ultrasonic wave or ultrasonic frequency. Now come to the After this ultrasonic generation, come to our next slide. The first transducer is your fluid driven transducer. So the fluid driven transducer are used for the production of the vibration. Okay, we have a ultrasonic frequency, then we use that fluid driven transducer to produce the vibration, and that vibration are at the range of that ultrasonic frequency by forcing liquid to thin metal blade, which can be used for mixing and homogenization system. We can use we can force the liquid to pass through the metal blade, and this metal blade are what they are used for the mixing and homogenization system. So this is about the fluid driven transducer. Second is your magnetostrictive transducer. It is made up of the ferromagnetic material. Okay, magnetic magnetostrictive transducer is made up of what? Ferromagnetic material like your iron and the nickel, and the frequency of this oscillator can be adjusted. And how can be it adjusted? By changing the capacitance of that condenser. Then magnetostriction generator produce their ultrasonic waves, which are very completely low, and their frequency are at the range of 20 kilohertz. So this is about the magnetostrictive transducer. So we discuss about two transducer, that is your fluid driven, and the second one is your magnetostrictive transducer. Next is your piezoelectric transducer. It is like a naturally piezoelectric occurring material is used for the production of this piezoelectric transducer, like your beryllinate, can sugar, quartz, rock salt, topaz, tourmaline, and bone. Okay, but this piezoelectric material can also be made by the man-made material. Okay, like your barium titanate, lead zirconate titanate, and the frequency range of this piezoelectric transducer, which can be produced by this piezoelectric transducer, is at the range of 300 kilohertz. Okay, piezoelectric transducer produce the frequency of that ultrasonic wave is more than about 300 kilohertz. And in application system, a coupled device is used to transfer that ultrasonic vibration to the sample, probe sample system, and ultrasonic bath. Next, this is your probe system, and the first diagram is show the probe system, and the second diagram shows that the ultrasonic system. Use of the ultrasonic wave ultrasound in a food processing. So we can use a ultrasound in a food production, which means that we use it for the conversion of the raw material into the product. Okay, like filtration and drying. In filtration, what we have to do, we remove the unwanted material. Remove the unwanted material. Unwanted means that the contaminated material. The contaminated material. Okay. And in drying, what we have to do, we remove the moisture content. So for both purpose, we can use remove the moisture contain. So for both the process, we can use ultrasound waves for this purpose. Like we can also use it for a freezing purpose, like we kept the food product at low temperature, like around -18 to -22 degrees Celsius. Then this ultrasound can also be used in a mixing purpose and homogenization purpose. Like in case of the milk, when we homogenize the milk, okay, then defoaming, crystallization of the fat and sugars, cutting and degassing. So all these food processing can be done by the ultrasound waves. Ultrasound waves used in a food preservation. The first one is your microbial inactivation. So we use a ultrasound wave so that the organisms present into the food product present into the food products can be destroyed. It can be done by the cavitation phenomena. Okay, now what is this cavitation from phenomena? In cavitation phenomena, there is a changes occur in pressure and temperature, and due to this changes, the cell wall of the microorganisms are breakdown, disruption, and thinning of the cell membrane and DNA will be damaged. Okay, when a sudden change occur in pressure and temperature, then the cell wall of the microorganisms are break down, and their cell membrane either it is disrupt or become a thin, due to which the DNA will be damaged. This is your cavitation phenomena or cavitation process. So this cavitation process can be divided into the two category. The first one is your transient cavitation. In this type of cavitation, the bubbles accelerate in a irregular fashion, due to which it is finally implode. Then a high local pressure is produced and high temperature is produced, due to which biological cell are disintegrate and the enzyme present into it is also become denatured. Then next is your stable cavitation, in which the bubbles are oscillate according to a regular fashion for many caustic cell. Then this inactivation affect the ultrasound has also been attributed to the generation of that intracellular cavitation, and due to which the mechanical shock occur, and that shock disrupt the cellular structure of the microorganism, and the functional component which are present on that microorganisms become cell lysis, means that the cells are disrupted. Method of the ultrasound. So it can be divided into the several method, the like your ultrasonication, thermosonication, monosonication, and last one is your monothermosonication. Okay, so ultrasonication are application this ultrasonic can are applicable for a ultrasound wave at low temperature. Okay, then it required a longer treatment time for the inactivation of the stable enzyme and microorganisms. That's why it require a high energy requirement. See, ultrasonication system is what? It is only application of ultrasound which have very low temperature. Okay, that's why it's required a longer treatment for the inactivation of the stable enzyme present into the food product and the microorganism present into that food product. That's why it required a higher energy for this process. Next is your thermosonication. So thermosonication are the combined method of the ultrasound and heat. When we combine the ultrasound with the heat, then it become what? It become the thermosonication. It produces subjected to ultrasound and moderate heat simultaneously. Okay, when we apply the thermosonication, so it produces the ultrasound wave. At the same time, it also produces the moderate heat, and this method is produce a greater effect on inactivation of the microorganism than heat alone. Okay, so when we combine the thermosonification, then it produces a greater effect for the inactivation of the microorganisms as compared to the heat alone. Then monosonication is the combination of the ultrasound and pressure. We apply the pressure and the ultrasound together into the microorganisms and enzyme, due to which it inactivate the enzyme and the microorganism. Then monothermosonification. In this method, we combine all the techniques like we combine the heat, ultrasound, and pressure. Okay, it can be used to inactivate these several enzymes at a low temperature, which get in a shorter time as compared to the thermal treatment at the same temperature. Effect of the ultrasound in combination with the heat pressure. So the first diagram, the first column is for the inactivation by, like either it is done by the ultrasound alone, then the combination of the ultrasound and the heat, then the combination of the ultrasound, heat and pressure. And the second column is for the vegetative cell, then third column is for the spore, then last one is your enzyme. If we use a ultrasound alone, then it can only affect the vegetative cell. Okay, there is no effect on the spores and there is no effect on the enzyme. When we combine the ultrasound wave and the heat together, then it can affect the both vegetative cell and the spore cell, but it cannot be affect any enzyme. There is no effect on the enzyme. But when we combine the ultrasound wave, heat and pressure together, then it can affect the both like all these three categories, like your vegetative cells, spores and enzymes. Microorganism inactivation. Microorganism inactivation. So we all know that there is a different kinds of the microorganisms are present and they have a different membrane structure. Okay, so there, if we talk about the gram positive bacteria and the gram negative bacteria, so gram positive bacteria have a thicker cell wall as compared to that gram negative bacteria which have a thinner cell wall. Here the cell wall means that the peptidoglycan layer, not peptidoglycan layer. Here we talk about the only thicker cell wall. Okay, so gram positive bacteria have thicker cell wall and gram negative bacteria have a thinner cell wall. So there are the several factors which affect the effectiveness of that microbial inactivation, like your amplitude of ultrasound wave, exposure or contact time, volume of food process, treatment temperature. So, if we have to inactivate the Listeria monocytogene by high power ultrasound wave that at the range of 20 kilohertz, then we required a ambient temperature and pressure, which have been found to be low with decimal reduction value 4.3 min minute. By combining sub-leather temperature and high pressure of 20 kilopascal, these decimal reduction value will be lower than 1.5 minute to 1 minute. Spore inactivation. So microbial spores are very resistant to the extreme condition like high temperature and pressure and even at high and low pH and mechanical shock. So the endospores of that Bacillus and Clostridium, they are very sensitive to extreme condition. We required monosonication treatment at 15 kilopascal for around 12 minutes for the inactivation of the 99% of the spores, which increase the amplitude of the ultrasonic vibration of the transducer and increasing the level of the inactivation. For example, if we require 20 kilohertz probe at 300 kilopascal for 12 minutes, sonication for 90 micrometer amplitude, then it inactivate around 75% of the spores, then raising the amplitude around 150 micrometer, which give a result around 99.5% spore inactivation. Then thermal temperature finally increase the treatment, which give a result in greater rates for the inactivation of the certainly at 300 kilopascal, which compared to the thermal treatment alone. Enzyme inactivation. So the study of enzyme inactivation by the ultrasound has received a less attention than a microbial inactivation. Means that the ultrasounds are very effective in case of the microbial inactivation, but they are very less active for the enzyme inactivation. Ultrasound create a continuous vibration and produce a stable cavitation bubbles. These bubbles collapse due to the extreme local increased pressure, that is around 100 Pascal, and temperature around 5000 Kelvin. So due to that high pressure and temperature, the secondary and structure are breakdown and loss, and the enzyme are lost. So application of the ultrasound temperature and pressure treatment, this model is used for the enzyme relevant to the food industry in model buffer system, which proved to be much more efficient than the heat treatment. For the inactivation, these enzymes are especially those which are more thermally liable, like your lipooxygenase and polyphenol oxidase. So these are the table which shows that the inactivation of the enzyme by using a heat, pressure and ultrasound treatment. The first, the first column is for the enzyme, then medium, then treatment, then effect on the activity. Effect of ultrasound on the food quality. So the first effect on a dairy product like serum protein are lactoalbumin and lactoglobin. They are denatured when an ultrasound is combined with the heat. Then with these two treatments performed separately. Similarly, result we obtain the fat globule homogenization when a continuous monothermosonication are applied. This also result in slight change in milk color. Then, what is the effect of that ultrasound on the juice? Vitamin C are retained of orange juices when we treat it with the ultrasonic treatment is higher when it is compared to the thermal processing. Okay, when we do a thermal processing, we lost that vitamin C, but it can be retained when it is subjected to the ultrasonic treatment. The effect of ultrasound and temperature on vitamin C content of tomato extract, it was found that there is no significant effect of the ultrasound, whereas heat treatment is significantly reduces the vitamin C content in case of the tomato extract. MTS treatment of pure pectin solution yielded the molecule with the lower apparent viscosity due to the size reduction. Conclusion: ultrasound is considered to be an emerging technology in food industry, and they have very advantages for minimizing the flavor loss, increasing the homogeneity, saving the energy, high productivity, enhance the quality, reduce the chemical and physical hazard, and is environmentally friendly. When it is applied with that pressure and temperature, its efficiency is increased and it control the nutritional loss. Ultrasound is good alternative method for the food preservation, and also no adverse effect on the human health. So this is all about the ultrasound as a preservative. I hope you will understand. But if you have any query related to this, you are free to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.